My name's Paul Ackerman. I'm a graduate student at the University of Colorado, Boulder. And I'd like to first recognize my advisor, Ivan Smiluk, as well as Yao van de Lagemont at the National Renewable Energy Laboratory in Golden, Colorado, as well as other Smiluk lab members who have helped me out. So today I'm going to tell you a little bit about my efforts towards using a liquid crystal defect as an optical nano-laboratory. And I'll explain what that means, but first let me motivate why studying nanoparticles is interesting. There's increasing energy demand, and so the world's energy infrastructure is going to have to change in some way. And so it's predicted that photovoltaics will end up playing a major role in this future energy infrastructure. Current technologies, shown in green, such as silicon-based photovoltaics, are very expensive to produce and don't have extremely high efficiencies. And so economically, they're a little bit not viable. Second generation photovoltaics, such as organic photovoltaic, shown in yellow, is recently showing that it's very cheap to manufacture these types of devices, but they really suffer from extremely low efficiencies. And then there's this third generation idea, such as nanostructured photovoltaics, that would allow us to have this cheap manufacturing, however, a very large energy efficiency. So this is what we would like to head towards, but there's still a lot of work that needs to be done in terms of understanding the microscopic properties of nanoparticles. So a nanolaboratory would really be helpful in order to take a close look at these nanoparticles and understand what's going on. And my work will be towards looking specifically at plasmon-exciton interactions. Like the last talk, this is a bottom-up approach where we're going to be looking at fundamental properties of nanoparticles in order to work towards an efficient device as opposed to studying device properties. So typically when I think of a laboratory, I think about chemicals, beakers, and fancy equipment that has knobs to adjust experimental parameters. So what I mean by a liquid crystal defect nanolaboratory, I mean a small volume where I can contain my nanoparticles, and then I'm able to adjust experimental parameters such as applying voltage and changing the orientation of the liquid crystal, as well as adjusting the heat and changing the size of the isotropic core of a singular defect. Here I show a schematic where you could imagine the isotropic core of a defect that by controlling temperature, if there's two particles in here, maybe they find the isotropic nematic interface, and controlling temperature, I'm able to adjust the separation. Then we can also probe it. We can image it with an objective, use laser tweezers in order to manipulate the particles. So this is just a schematic of what the idea might be. Particles that are interesting for photovoltaic applications include plasmonic particles and semiconducting particles. So here I picture different types of gold nanoparticles and the nematic director structure that would surround them if we disperse them, say, in a thermotropic. And these particles are interesting because they have surface plasmon resonance. And this is a collective oscillation of their electrons, which can enhance the optical field around them many times. Makes them a very interesting thing to use for experiments. Then the semiconducting nanoparticles are used for labeling and bioimaging and biosensing. And however, these semiconducting nanoparticles all display some sort of fluorescence intermittency. And this is a property that's not fully understood. However, to be able to use them efficiently, to be able to create a third generation nanostructured PV device with these particles, it would be really nice to better understand all of the microscopic physics going on here. So these are the particles that we're interested in studying. Now, you guys have heard a little bit about defects in this school. Here are some pictures of defects. Liquid crystal defects are typically characterized by a region of reduced order. And so here is a schematic of this so-called toron, and the double twist cylinder looped upon itself, 2 point defects in order to meet surface boundary conditions. And I've shown that I can draw them in arbitrary patterns as well as linear defects. And this is just demonstrating the control that I have of these defects in the liquid crystal. Defects also can be stabilized through the inclusion of colloidal microparticles. So for example, here we have colloidal microparticles that have stabilized some sort of point defect near their surface. And then we've used this to sequester a nanoparticle in that point defect, and then used optical tweezers in order to assemble these superstructures. And these are just good examples of the control we have of our beakers. And so I show a video here. They're a little difficult to see, but there's a twist disclination in a confined cholesteric. And so the nanoparticles in the area that have not stuck to the substrate have found their way into this defect, displacing some of the high-energy melted liquid crystal. And so they get confined in one dimension, and you see them dancing around due to Brownian motion. And this is just a movie of something kind of interesting. And then also I show here, there's 2 torons, and the one now in the center, you can see a nanoparticle that's confined in one of its point defects. And now it's confined in 3 dimensions, and we've also done experiments to show that there's somewhat orientation of a elongated gold particle in these defects. So the idea is there's photo conversion processes such as multiple exciton generation. I show a little schematic of what this is. A photon comes in, and if the photon is at least twice the band gap of the material, then there's a possibility of having a coulombic interaction, which causes charge multiplication, and we get twice the current than we could have with, a photon of only once the band gap. So this would be something we would like to take advantage of in our nanostructured photovoltaic. However, these particles have this fluorescent intermittency, the blinking, and so here's just a graph that's supposed to represent what this means. There's periods of time in which the particle is fluorescent, and we can continuously excite electrons and then have them fluoresce. However, their electron can be stuck in, or the particle can be stuck in a dark state, and then, well, people don't exactly understand all of the physics here, and so hopefully I'll be able to use a liquid crystal nano-laboratory to study it a little bit more. Here's a schematic of example of the setup that I've been using. We have scanning mirrors we can bring in a laser for imaging or exciting the particles. Then it's an inverted microscope. Then I can use some photon counting hardware in order to look at the amount of signal coming from the particle that I'm exciting. And I'll just show some results that I had recently. I'm looking at a fluorescence trajectory, and maybe it's a little easier. I can make a histogram of this data where each bin is about 10 milliseconds, so it's the number of photons I count in 10 milliseconds. Make a histogram of it, and you see 2 states, an off state and an on state, very typical of this blinking. And so that's an example of one of the things that we can start studying, these nanoparticles, once I can control them in the liquid crystal defect. However, here's another twist disclination, and I want to show taking laser tweezers, and I'm able to push these two gold particles along the disclination towards each other and actually get them to appear to touch eventually, and thinking about including semiconducting nanoparticles between these two plasmonic gold nanoparticles, be able to look at the changes in the blinking statistics and maybe be able to back out some cool physics this way. I also show a dark field image sequence of a particle being pulled into the point defect of a toron, as well as the toron with the particle in between cross polarizers in a bright field microscope. These systems, we can polymerize them as well. So in C and D here, I show the system after I've polymerized it, so I include some sort of reactive mesogens to the liquid crystal, and then polymerize it so we can lock in configurations that we're interested in, and this just makes it easier to work with or possibly be able to make a stable configuration. And then the schematic of we are able to image this gold particle that is stuck in the point defect of a toron and determine that this rod-shaped gold nanoparticle was oriented vertically in the defect. So this is what I've done so far, and here's a list of recent publications that are relevant, and I guess that's it.
You have talked about the gold nanoparticles, and especially the optical phenomena can be enhanced by gold nanoparticle. You have talked about the SPR, surface plasmonic resonance. Do you think that the SPR are localized or non-localized?
So,
localized SPR or non-localized SPR?
In the particle.
Yeah.
Well, I actually don't know the difference. So, I think the surface plasmon resonance for these particles is well, these particles are that I've been using have been tens of nanometers in size. I'm not sure if that helps answer your question.
So you have additionally used semiconducting type nanoparticles?
Yes.
Which one?
So we've been using cadmium selenide.
So what do you think, that it can also enhance optical properties or not?
Oh, I think it definitely can, and I've observed it just so a gold particle next to a semiconducting particle, you can see just with your eyes. Well, maybe you don't want to look into the microscope, but you can see that the semiconducting particle does not have any visible fluorescence. You bring in the gold particle, and all of a sudden you can visibly see the fluorescence from the semiconducting particle.
But if the optical enhancement is due to the semiconducting nanoparticle, then SPR may not be hold there.
Well, it's something that it would be interesting to look into using one of these.
Because in semiconducting type of nanoparticles, mainly a depth type of resonance occurs there. Donor acceptor pair resonance occurs there because electron and holes both present together. So maybe.
So you're talking about quenching. So if you bring the particle in too close, of course you're risking quenching the fluorescence. But if we can tune the separation between two particles, then we could explore this regime and actually understand it. I think that there have been some studies done actually using AFM tips to figure out the separation that quenching actually begins. But this might also be a neat situation where it's actually in solution. So it's interesting.
Thank you.
Yeah.
One more quick question.
How do these plasmon excitations depend on the fact that the particles are actually in the defects? As compared, say, to the previous talks where you had just, say, a dense suspension of them.
So this is something a little bit complicated since you have a spatially varying index of refraction. It's something that maybe some models would help illuminate, but some simulation. But I guess I haven't put too much thought into that yet.
Okay, thank you very much.